Greetings, grade 12s. So, much like I did with the DNA section where we started on protein synthesis, this is now the meat and bones section of genetics, and that is dealing with monohybrid crosses and Punnett squares. So, this is really the more applied section that we're going to start moving on to. So please make sure that you have gone through the terminology videos, you have an understanding of that terminology, because otherwise you really are going to get lost and be left behind. Because you need to have a strong handle on words such as heterozygous, homozygous, dominant, recessive, all those kind of things. You need to make sure that you have an understanding of those words. So in the first video that I'm going to start with, I'm going to be dealing with autosomal disorders. So just based on this first page, what do I mean by monohybrid? So the clue is kind of in the name. So a monohybrid cross is where you have a combination of two individuals who are different based on one gene only. So that comes from the name mono, one. And the hybrid, two different individuals that are coming together. So they are, everything else for our intensive purposes is exactly the same. It's just the one gene that we are looking at. We keep it nice and simple for you guys in matric. We don't go into something like dihybrid crosses where there's two genes that are different, because then it becomes a lot more complicated. You can have trihybrid, and essentially with us, you know, there are lots of differences in a normal human, but just so you get a basic understanding of how the genetics works, we deal with monohybrid crosses. So remember, you are only dealing with monohybrids. At the same time, please remember, based on our Mendel video, we are going to use the law of dominance. There is no co-dominance in any of the things that we do, so if there is a difference between the two genes, one will be dominant, one will be recessive, and the dominant will always mask the recessive in this case. Yeah. Second thing from that front page, what do I mean by autosomal? So please, now, differences in terminology, don't get confused. Somatic cell is a body cell, a diploid cell, one that has two sets of chromosomes. For example, in us humans, a somatic cell will be 46 chromosomes. A gamete is then a haploid cell with half the number of chromosomes. So in humans, that would be 23. Now, the actual chromosomes themselves, we can categorize them into both autosomal and gonosomes. So an autosome is anything where the gene lands on, or its locus is on chromosome 1 to 22. So remember, us as humans, we have 23 pairs of chromosomes. So any of the genes that we deal with, that fall on chromosome 1 to 22, is going to be an autosome. It is essentially anything that is not a gonosome. What's a gonosome then? It is chromosome 23, the sex chromosome, your X and Y. So anything that has to deal with X and Y, that is linked to sex-linked disorders, anything like that, that is completely different. We are going to do a completely different section relating to that. So just know autosome is anything chromosome 1 to 22. At the same time, note for yourself, when you are dealing with autosomal things, you are only using letters. There is no X's and Y's here. So it'll make sense when we get to the sex-linked video, but just note we are only using letters. Okay, let's get into it. Scenario 1. A purebred black rabbit is crossed with a purebred white rabbit. Black is dominant. What will the offspring be? So, first of all, what is the gene that we are comparing, or what is the gene that is different? It is fur color. So I've got a black furred rabbit here and a white furred rabbit here. And I have told you that black is dominant. Sometimes we don't tell you which is dominant or recessive. Sometimes you have to work it out. But step 1, because we know these things, we have to assign some letters to the different alleles. So let's say that let capital B equal black fur. So capital B is going to represent black fur. And then we are going to let small case b represent white fur. Now please note in this, I have used the same letter, but they are different sizes, or they are different, like caps lock versus lowercase. I don't use B for black and W for white. And the fact that I use B, I can use any letter. It doesn't have to be related to the actual condition itself. Remember, this is a variable. You can assign whatever you want to it. So, but you must keep the two letters the same. You can't have a B and a W because then you are showing different genes because they represent different things. As a side note as well, if you are to choose what the letters are, sometimes you do get told, use this and this, but if you have to assign it yourself, choose letters where their capital version is completely different to their lowercase version. So something like S and O and C, those kind of things, their capital and their lowercase are very similar. So don't use those. Rather use letters like A, B, T, Q, F, things like that. You know, you can use what a P sometimes is an okay one, but even then, make sure that the differences between them, so at least you don't get confused, and that me as the examiner doesn't get confused between what you're saying. So, what we're going to do is we're then going to introduce this thing called the Punnett square. And literally, it is just a square of 4 blocks. So you will have to draw this square as part of the process that we're going to do. So you would use a nice little ruler, make it nice and big, don't make it nice and small. Really make sure that we can see the square that's here. So I told you in the question that we had a purebred black rabbit and a purebred white rabbit. In this instance, what does purebred mean? Purebred is then referring to homozygous. So remember we said we've got capital B and little b. What then the parent genotypes of the two individuals that are becoming the hybrids, what is their genotype? It will be, for the black rabbit, big B, capital B, we refer to big and small, because it is a homozygous black. And the other one will be little b, because it is homozygous white. So we have to write this as our first thing because we are identifying the parent genotypes. This is part of the question. It's part of the skill that you need to know. So we normally put a little X in between. So this X is not multiply as in maths. This is showing is crossed with. So this genotype is crossed with this genotype. Okay. So we have identified the parent genotypes. Remember, this parent inherited this from potentially its mother and this from potentially its father. One from each of their parents. And they inherited one of each of their parents. Their offspring will only be able to get one of these. So the offspring of this merge cannot have both of these. Because remember, these get separated into gametes. Law of independent assortment, law of segregation. We need to separate these from the parent. So, as part of the first step, we need to separate the two gametes, or the two genes. We separate the two genes. So based on the 2 points of the square, I write one at this square and one at this square. And then I do the same at the top for the other one. I then separate the little b and the little b. So now I have my separate genes. They've gone into their separate gametes. Now I need to show how are the different combinations going to be represented. So then I need to combine them. So how I do it is I read it much like how we did with when we did the mRNA coding table. I start with this end. So where this one and this one meet in the square, I must write what is going to be the combination. And that would be a big B and a little b in this little block. What would this block be? Well, whatever links up from this side and this side. And it's going to be also big B, little b. What's going to be in this spot here? Well, it's going to be the same. So we kind of notice what's going to happen here. And what's going to be in this block? I then have that. So this essentially is the drawing of the Punnett square. Okay. So, what do I do from there? So I then take the things that are inside my Punnett square, and I have to answer several questions. So question 1, what are the possible genotypes that can occur from this union? From this hybrid, what are the potential genotypes? Well, if we look back at the picture, what are the potential combinations that we can see? Well, clearly, the only one is big B, little b. There is no other combination that can occur. It can't be big B, and it can't be little b. Those combinations just cannot occur. So that's the first question. Then, what are the possible phenotypes? So remember, genotype, what are the specific alleles that you have inherited? Phenotype, what is the physical outward expression of this? So if these are the potential genotypes, what are their fur colors going to be? And this is where we use words to describe what's going to happen. They are all going to have black fur. Because remember, we are dealing with the law of dominance. This is a heterozygous combination because they are both different. One will be dominant, one will be recessive. And I told you in the question that black fur, what we chose to represent by big B, is dominant over white fur. So therefore, if it is present, even if it is heterozygous, it will always be expressed in the phenotype. Remember, we deal with complete dominance only. Okay. So I'm going to leave that as part 1 of this video. I'm going to go part 2 of this video a little bit more in depth. Go back, see the steps that I went through, because we're going to set out the steps of how we physically answer a question relating to a Punnett square in the next video. So go through this. If you're a little bit confused with the terminology, make sure you understand the terminology, and then we'll come back and we'll get running into part 2. Thanks very much, guys.